Okay. Hopefully you can see this. Thanks very much for joining us. So this is a meeting that I've been running for a couple of years now, called Balti and Bioinformatics. And the reason we call it Balti and Bioinformatics is because we talk about bioinformatics, and then we go for a Balti. And I realise many of the viewers may not know what a Balti is. This is a Balti. A Balti is just a curry, really, and it's served in a bowl, a traditional bowl called a Balti. And that's a specialty of the Birmingham region, so that's where I'm from the University of Birmingham. This is a special Balti in bioinformatics because obviously we're not having a Balti because we're online, but we will be going for a Balti afterwards, and you're very welcome to as well. I just want to check that anyone's out there firstly, so if you want to talk to us, you can communicate via the Balti and Bioinformatics hashtag, if you want to ask us questions, if you just tag it with that hashtag, we should be able to see it and hopefully respond. So we've got a very exciting program today. It's starting with Clive Brown from Oxford Nanopore Technologies, and you'll spot a very definite nanopore theme to this session. After that, I'll be talking about some of our experience with nanopore data, and then Matt Luce from Nottingham University will be talking about live streaming nanopore data, which is very apt for this session. Then Josh Quick from my group talking about a real-life application of nanopore. And finally, we have a visitor, Torsten Seemann from Monash University, with us in Birmingham for most of the month, and he's going to be talking about his pipelines for microbial genomics. So this feed is slightly delayed. I can see that there are 85 viewers online. I'm hoping that that you can see this. I'll just put a quick tweet out and just check. Is that okay? Okay, great. Perfect. All right, so without further ado, we'll get on with our first talk. And I should just say, you are free to tweet the contents of this talk. Obviously, it'd be crazy not to. We will try and answer some questions after the session. Some of the talks that the presenters may prefer you not to tweet, in which case they'll say, and I'd be grateful if you could respect that. Not all of the talks will be saved for posterity. Most of them will. So you're best off watching it live, but some of the talks at least will be saved on YouTube if you want to go back and refer to them later. So over to, hope you can see now Clive Brown on your screen. Clive Brown is the Chief Technology Officer at Oxford Nanopore. Some of you may have seen him before. I'll let him introduce his talk right now, and over to you, Clive. Thank you.
Thanks, Nick. I hope everybody can hear me. Can you hear me, Nick, clearly?
I can hear you clearly.
Great, thank you. Now, I haven't done this before, at least not for a long time. Normally when I talk, about 5% of the audience fall asleep. Even in an Oxford Nanopore board meeting, I can put people to sleep. There's something very soporific about my talking style. So with half an hour to run, and a very large number of slides, I might have to skip a number of things and focus on others. And I'll be watching Torsten in particular to see if he starts nodding off while I'm talking. So let's see if I can get this thing to run. Now hopefully you should all now be seeing slides. Is that right, guys? Can you see slides?
Yep.
Yep, great. Okay, now I have to cover some background because I imagine there are people watching who know very little about nanopores, and I apologise if that's boring for people who know a lot about it, but it just has to be done. Oxford Nanopore used to be a fairly unknown company. We've been around for quite a while. You may know that we now have going on for 200 employees. Quite a recent expansion. We've raised a reasonable amount of capital, over £180 million over the past several years. And we have a very large IP portfolio licensed in from collaborators, in particular Harvard, Oxford and UCSC. And a little known fact is we continue to fund several $1 million worth of academic research per year. Now, we are a nanopore company, the clue is in the name, but that concept is actually quite old. Certainly the IP goes back about 20 years. And I first heard of it in around '97. And one of the authors on the key sort of foundational piece of IP kindly sent me a page from his notebook that he made at the time they had the original discussions around nanopore sequencing. And here it is, and you can see the concept is certainly for the version that we work on, which is called STRAND. It's very simple. A strand of DNA moves through a channel, and that channel is insulating. And ions move through, and we detect current. And he sketched out a picture of what he thought the signal would look like at the bottom there. Now, it's taken really 20 years to get to where we are. An enormous amount of academic research, an enormous amount of cross-fertilization in that research, and a lot of government money, particularly in the US, has gone into this. Now, there is on the website just a history of nanopore. I'm sure most people aren't interested. But in essence, we got our proof of concept data in late 2011 on DNA sequencing with STRAND. For various reasons, we announced that in 2012, and we announced our intention to launch products, and we described what those are. And then it's in 2014 that we've actually begun our early access program in earnest. Now, the device that's out, the one most people are familiar with, I'm going to focus on today, is called MinION, universally mispronounced, and an awful pun, and of my invention, so you can blame me for most of the company naming schemes. Now, MinION is a USB DNA sequencer. It is obviously portable. It's designed to plug into a laptop. Of course, it's obviously radically different from any of the DNA sequencer on the market. And we first had the idea to make this also in 2011. And when we sort of mooted the idea to make it at AGBT 2012, well, I thought it would be of interest, but I didn't think it would be of that much interest. And in 2012, we decided to focus on this as our first product. Now, obviously, it looks very simple, but in order to accomplish that simplicity, an awful lot of fundamental research has gone into what's inside that little metal case. There are two parts to MinION. At the moment, there is a reader, which is the sort of instrument part, which is the metal case, and then there's a plastic consumable flow cell that goes inside that. And the original product concept was to have it all in one unit, and we may circle back to that in future. But for now, at the kind of volumes that we're dealing with, it's much easier to make it in two pieces. Now, the heart of this sequencer is what's called the ASIC. That's an application-specific integrated circuit. And in a way, it's the processing core that translates chemistry into electronic signals. And these things don't grow on trees. There's no off-the-shelf ASIC that I'm aware of that's good enough to make an arrayed nanopore sensor. What most academics work on is the picture in the top right of this box here, which is a thing called an AXA patch, and it's really a sort of 70s-looking piece of electronics with some cables that go into a salty bath, where you measure tiny currents on biological things, whether it's nanopores or neurons, typically $30,000. And really the gold standard in signal to noise is set by the sensing circuits inside the AXA patch. And the main challenge for us was to co-design, actually with a company in California, we co-designed an ASIC that makes an array of those circuits in miniature. And those circuits are on a two-dimensional array. And really what they have are low noise amplifiers and analog to digital conversion, all on chip. And again, that's a significant challenge. And the signal to noise that we get on that ASIC is pretty close to the gold standard AXA patch. And I just emphasize that wasn't considered possible at the time we embarked on this program. Now, obviously, this is an ongoing area of research as well in terms of improving the ASIC. But really, you can't do high throughput nanopore sensing without a chip. Now, what is nanopore sensing? Well, the nanopore is just a very small hole. And that hole is, as shown here, we use proteins. Just point out, we also do solid-state nanopore research in the background. We don't talk about that much yet. But for the most part, our products are based on protein nanopores. And these things are ubiquitous in nature. They're everywhere. Your skin is covered in them. Most of the bugs that live on your skin secrete these things. And it's just a tubular pore that likes to sit in a hydrophobic membrane. And they self-insert at some frequency, which makes them nice. And the membrane itself, typically, there's a membrane, and on each side of that membrane, not shown here, we have electrodes. And the electrodes apply potential, a bit like a car battery. But the ions can't flow to the electrodes because the membrane prevents that. It's what they call a giga-ohm seal. It's insulating. When you punch a hole in it, that hole becomes a channel through which ions can flow. And when those ions flow, the ASIC can detect a change in current caused by that ion flow, as shown here. Now, so here's a sort of schematic of a typical nanopore signal. An open pore, as we call it, the open pore current is just a flat line here with some fuzz, which is noise, sensing noise. When an analyte interacts in the hole and blocks it, as you might imagine, you see a downward deflection because less current flows through the hole. And that deflection will have a period, a time that it will last for. And those times can be very quick. Most interactions are femtosecond. So most people in this field have to engineer the pore to give specific long-lived interactions with certain analytes. And what that means is the system can be exquisitely discriminating for certain analytes. So really we talk about block and dwell as the two main characteristics of a nanopore signal. And then on that signal we have some noise here, which is this fuzz. Okay. Now, some time ago, some papers came out really in sort of 2010, 2009. These are worth reading if you are interested really in getting into this. And these are the things that made us decide to focus more on strand. And it's Mark Akeson, who's a collaborator of ours at UCSC, who showed that he could, in solution, bind a polymerase to a double-stranded piece of DNA. He could catch that on a pore, and he could generate a somewhat reproducible signal from it. I'll just wind back a little bit. DNA itself, as an analyte, is negatively charged, and it wants to go through a pore when you apply potential. It will zip through. In fact, it will zip through at 1,000,000 bases per second. So really the challenge here is to trap it and slow it down. This is how Mark did it. Now we reproduced this work very quickly, which is very nice, but we quickly found that the movement of a lot of enzymes just simply isn't good enough. They tend to move backwards and forwards and hop and skip. And if you can't get unidirectional, one base at a time movement, it becomes nearly impossible to interpret these signals. Now Hagen and some others showed that if you take a piece of DNA and dangle it in a pore, and he just used a big blob of protein, and again that will just get trapped and sit there as a static complex. But he showed that by modifying the inside of the pore at the narrowest point closest to the DNA, he could change the block of that DNA in a way that appeared to be characteristic of certain bases. Now again, we reproduced all of that, and we reproduced it in both a static and a moving system, and we found that pretty much every protein pore actually measures more than one base. It measures 3, 4, 5, even 11 bases at once. And that when you change that protein, you actually can change the signal given by a collection of bases. So our challenge then was to make it better and to figure out how to decode those signals in a reproducible fashion, which of course we did. And that was our proof of concept. Now nanopores, again, not widely understood. The array sits there with pores and they're empty, and there's ions whizzing through. And nanopores take analytes from solution, very different from other sequences where you typically have to attach the DNA to the sensor in some way. We don't do that. They're all swimming around in solution. And again, naturally, DNA will get sucked into the pore. It's like a black hole. It gets within a certain distance of the pore and it will zip through. And so here's DNA with a protein bound, a break protein. And that break protein will then slow the passage of the DNA through the pore and unwind the opposite strand. And only certain proteins can do this. We did an enormous search for the right proteins. Now in our case, we don't have to do this, but we do. We put a hairpin on the end of the DNA, and then the system will process around the other strand, and we can get both strands of the DNA sequenced and readable. And when that's finished, the whole complex dissociates and the pore becomes available again, it becomes open to accept a new molecule. So the system is constantly sampling from solution, and it's a streaming system. It's free running. It gets very different from other sequences that I'm aware of. Now that of course occurs in parallel on the ASIC, and underneath each pore, a circuit is detecting what that pore is doing. But again, a key difference here is there's no synchronization between the channels. They're all running at their own speed, and they're all running in a way that there's no sort of coordination. So what that means is at one time some pores have DNA and at one time some don't, and every pore can have different bits of DNA sampled from solution. There's no concept of a cycle on this system. Now obviously that is very different from conventional systems, and it means that we get full-length reads in real time. That means that a pore will process a piece of DNA, and that piece of DNA is available for analysis immediately. There's no real concept of an end-of-run analysis step, unless you want to do it that way. Now obviously MinION is designed to come with a shippable pore array, and another thing that people said was impossible was making a shippable pore array, and that's because most of the academic work done to date has been done on Axopatch on lipid bilayer systems. Again, we quickly found that wasn't good enough, so we had to come up with our own system. And the way we did that was twofold. We use a we put a micro-patterned surface over the top of the ASIC, such that over each electrode there is an individual well, as shown here, and there's some very special patterning on top of the wells. And then we sloosh in a sort of mayonnaise of buffer and a membrane material in oil. And the system is designed through the use of surface tension and viscosity and other physical parameters, and the shape of the patterning is designed such that those reagents self-organize and separate out on the surface, such that we get water in the well, as shown here. Here's the electrode. And on top, we get a thinned out membrane of that special material. And here it is in section. And the pore will then insert in the thinned out section, which might only be a few nanometers thick. Now all of that system, coupled with the membrane material itself, which is not a lipid, gives us very stable chips. And those chips equilibrate. If you make them equilibrate, they will last a very long time. And that should be apparent by now. You can FedEx those chips to people all around the world, and the pores are intact and they're runnable even for several days. So that is obviously another key thing you have to do with protein pores. Another thing we had to do was get the on rate of the pore high enough. So we want the system to be very sensitive, and the initial approach we've taken to that is to enable users to tether, we call it tethering. That means you can absorb the sample to the membrane material, like a lawn. Now, as far as we can tell, the molecules are mobile. That's a 2D membrane, and it's mobile. And that means there's a 2D search going on here. And the on rate, then, of any molecule is about 10,000 times higher than if we're sampling from solution. And that tethering technology is reversible. There is an equilibrium there, and there is a wash-off step that can strip everything off the membrane. And that tethering can also be specific. That is, we have versions of it not yet in the field that let people target certain bits of a genome. In other words, you can hide and select in situ. And another key point to make there, again, I think it's very significant, it's not often understood, is there's no PCR in the system unless you want to. And I think it's the only system that directly interacts with the analyte, the DNA, and reads everything that's there, warts and all, quite frankly. I think every other system reads a 4-base complement. Now, okay, we can speculate on the utility of that. I think it's going to be very important in the future. But certainly we see damage, we see base modifications, and we see the 4 bases. Now, okay, we had to go out with a library prep, and the one we've gone with initially is on the right-hand side here. It's pretty simple. It's a G-tube-based prep. Here's your DNA, and the system does a an adapter ligation and a repair, and the hairpin goes on one end, and the other end is a pre-packaged leader, and we have the enzyme on the leader. Here's the tether. Now that's expected to take about 2 hours, but actually in a skilled person's hand it's more like an hour. And in our proof of concept work we showed another system where we used a transposase, as shown here. And actually what we do now is we can load up the break enzyme on the transposase. We can preload it into one complex. And in a one-step reaction that can now put the requisite ends on the DNA and put the enzyme on. And that is down to minutes. And in a way, as I'll cover later, it's 0 minutes. But that's coming along towards mappers as we speak. Now, Great Place made a read length, and again, early on in the system, we found that read length is, as far as we can measure, sample prep dependent. So if you make a distribution of fragments in your G-tube and you measure it on an Agilent bioanalyzer, and then you measure it on a pore array, you pretty much see the same thing. Certainly, that's what all of our standard test samples show. And so read length means are quoted really from that, and it's really what the user puts in. And we've certainly seen on the map people trying preps that give longer fragment sizes, and they get longer fragments in the array. And there's no reason to think the pore can't process enormous fragments, actually. I think there is a natural tendency for very large bits of DNA to start breaking, so there may be some physical limit. But I don't think anybody's really reached that limit yet. Now, if we look at if we look at a fragment distribution from the sort of standard shear, we get something that's basically Gaussian. And if we look at the transposome prep, we get something that is skewed. So the latest version we have, which again is in kit prep, we get around a sort of 10 kb mean in this particular case, and then a very long tail going up to 50, and in this case it's on lambda DNA, so the maximum possible is 50. And I don't know what would happen on a bigger genome. You might get a much longer tail. So in our view that's good enough, and we're going to throw that out to people to play with very shortly. So really sample prep is actually fairly trivial, and I think it can be a lot easier, and certainly that's a key area of focus for us. And on that theme, even the G-tube version is about to get streamlined. Again, this is a little insight into what's happening in MAP. We think we can get the G-tube sort of standard random shear kit down to just 2 tubes effectively. We think it'll be effectively a 2-tube kit with some accessory control DNA. So I think even that's going to get simpler. Now, okay, of all the things we said were impossible, we've dealt with most of them. So what we're left with now, most people focus on base calling and accuracy, so I'll spend some time on that. So how does this thing work? Well, in essence, it's a machine learning problem. Because we found very early on that a given piece of DNA will give you a very characteristic signal that's very, very reproducible. So in essence, if you come up with a way to learn those sequence-specific patterns, you can use that to decode new signals from other fragments. That's all there is to it. So our raw sort of signal looks like this. Open pore is up here, it dips down, here's the DNA going through. And for historical reasons, we have names for these things. We zoom in, you can see the raw data here. And this sort of spiky fuzz is because we measured very, very quickly. A key point about the ASIC is that it samples very quickly, because we don't want to miss anything. Now what we do then is we put a step weight through that. There's a little algorithm that does that, and that's quite a big data reduction step. And we take, effectively it's a mean, and we take out the time domain, so we take out the dwell. Now you can argue whether there's information in dwell. Not an area that we've put an enormous amount of work in. You can argue also if there's other information in the noise, and certainly in some of the earlier chemistries we saw information in the noise potentially, but at the moment none of that's in the sort of core base caller. And what we end up with then is this blue line here, and this is called a squiggle, for want of a better word. Effectively it's just a trace, and the shape of that trace will be characteristic for a piece of DNA, and that's what the base caller has to deal with. So that's what our base caller deals with, but there are other schemes possible, I believe, that can work on these other levels of data. And I know people on MAP are developing those quite actively. So how does it work? Well, it turns out that a pore gives you a level from a k-map. So every pore has a sort of list of k-mers with a block level that's characteristic for that k-mer. Now, when the enzyme moves one base, and this is why movement is so key, for any given k-mer, for example here TAGGG, there are only 4 possibilities. There's only, well, in a 4-base scheme, there are only 4 possibilities I can append to that word. So we're looking for a transition that has AGGG and one of these 4 bases on. So really there's a choice of 4 places to go, effectively. Now, if you look at a long enough stretch of squiggle, what the base caller has to do is, with its pre-trained idea of what k-mers map to what signal, it has to find the likeliest path through that signal and emit the likeliest sequence. Now, there are lots of ways of doing that, and my guys have implemented a version using HMMs. But I've no doubt there are other schemes to do it. And this is not a particularly novel, well it is, but it's not, these kind of schemes are not that unknown in the world of signal processing. I'm going to speed up. Now we can also do both strands, and of course the signal on the complement is different and complementary to the signal on the template, and those can be combined, thus boosting your ability to base call and even detect damage and modified bases. Now here's a squiggle just showing, it's a real squiggle, it's just showing this scheme. DNA goes in, here's the template. We put a marker in the hairpin, we take out some bases here. The easiest base to see is an A basic, i.e. nothing there, because it's a hole. And so we see this enormous spike, and then we're transiting through the complement. And again, the latest base caller can combine those data. And again, very often issues in the strand can be clearly complemented in the signal from the complement. Now here is an R7.0. So people on who aren't on map don't know this whole story. I'm not sure I have time to go into it. But we've updated our chemistry recently. From initial chemistry that went out with a number of issues, we've updated it to a more recent version. And here's another typical read, and from user reagents. And what we tend to get are chunks of good data interspersed with bad data. And there is a base scoring scheme on these data. You can a priori identify good and bad bases, so I'm not sure it's widely exploited yet. And on the version of the base caller that's in the field right now, this would be the typical distribution of 2D error rate, counting all errors, for want of a better word. Now, I don't like talking about this because if you parameterize the base caller differently or you change a few things, you can move these numbers around quite dramatically. And more importantly, the base caller we have, which is available to mappers, is not fully feature functional yet. In other words, we haven't put in all of the whistles and bells. And mappers who've been digging into these data, if you go and look underneath these bad bits, you will see signal. And you will see sequence-specific patterns in those signals. And I know that people have been implementing error corrections and things. So there's still, I think, a significant uplift just in software. And here's a slightly more recent chemistry that went out last week. Some early data coming through. Again, they're not a typical read. Tuned up a little bit with base calling and whatnot. Now again, it's very different. The alignment parameter is different, and you effectively have an enormous sort of multi-KB read with good, a little bit bad, good, a little bit bad. And that's how the data is as it stands right now. But what I would emphasize is we have not hit the hard limit on data quality, for a reason I'm going to quickly move on to. This is just showing that pathfinding. So here is the algorithm finding k-mers through that block space. Now, I think this is our data. I get confused. It might be a mapper data that we've got, but I think mappers have tweeted similar things. When you align these data correctly, errors average out in consensus, and I think most people have got high consensus accuracy at relatively modest levels of coverage. That's not to say there aren't some biases in the errors. I mean, most early platforms have some error bias, but it doesn't dominate the consensus, even at relatively modest coverage. And I'm pretty confident that that's going to be removed with future tweaks anyway. Hopefully people are writing those things up. So what's going on there? So where does error come from at the minute? Well, actually, a significant amount on our internal runs, a significant amount just comes from what I would call unmodeled data. Now, we know what that is and we know how to get at it, but it has not yet been rolled out into the public base caller in a way that's robust and self-parameterizing, etc. But it is being worked on quite actively. So I anticipate some software updates there will quite significantly deal with unmodeled aspects in the data. Because it's an early system, we've had quite a lot of heterogeneity in the things that we've shipped. Non-typically, when you send something out the door, you get spread and you get heterogeneity and contamination. One of the enzymes went off. And those have impacted a lot of the early runs. But I think with the latest releases, those are largely fixed, and people are catching up with our internal performance. And then, again, automatic parameterization of the base caller. It's a large area. But for example, because of that spread and heterogeneity, very often the event detection will fail because it's been taught to deal with quite good data. So that needs fixing up. And it's worth remembering that if we have a damaged base, say, or an error, it will affect about 5 bases because we're reading k-mers. Now, people have shown, and we know, that if you PCR your sample, you can reduce the error rate, probably because you're healing over damaged bases that are not yet in the model. So again, there's still, we're nowhere near a hard limit on data quality yet. I anticipate that gradually improving over time. An example here, I'm going to go really quickly, this is key. So the model we have out there, the thing the base caller recognizes, has a large amount of spread. It's an average model. But if we look at a k-mer, this is lambda, if we look at a particular k-mer at different positions in lambda, you can see that the signal is very highly reproducible. And yet, there is some deviation, so some local specific deviation caused by those contexts. And what that means is these are small, unmodeled context-specific differences that we have not yet built into the base caller. The base caller at the minute thinks that that k-mer looks like that. But even so, we get the kind of data I showed earlier. Another sort of error code, well known to us, is that the HMN again has its own sort of global parameter settings. At the minute, it will tend to hop over unrecognizable data, whatever its cause. And previously, we implemented schemes with k-mer specific transition probabilities that have somewhat ameliorated that. Again, those are not implemented in the field base caller yet. As I mentioned earlier, we've had some issues with manufacturing tolerances, and that can add noise to raw data, and that can throw off the event detection. And again, that will cause some degradation in field. So all those things are being worked on, and I would like to think the latest R7.3 and the latest tweaks should get most people a lot closer to where we are internally. Now, throughput, very quickly now. So where is MinION? People often don't realize that nanopore systems are actually quite high throughput. You can calculate throughput, as you can with a Selectra Illumina system, by the speed of the DNA moving through the pore and the number of pores on the array, and of course time. And there may be some other loss there, like duty cycle. And the minute we operate in this kind of zone, which is our best runs are around a gigabase for MinION. I think it can get up to a few gigabases in its current form. The next generation of MinION that we're dealing with, I think will go up into the tens of gigabase region, such that you could probably do a human genome at reasonable coverage. So the scope for expansion, both in speed and number of pores, and actually the numbers get very big very quickly. A surprise bonus to us was that we can make the chips very long-lived. We can actually make quite long runs that give quite large amounts of data. So there's upside also in terms of the chip lifetime. Now, duty cycle, very quickly, again, this is the internal data. Mappers don't get quite close to this. We get about 80% of pores sequencing 80% of the time. I think the best mapper runs are about 50% at the moment. Again, there are good reasons for that. And I think we've had up to gigabase on our systems, and I think the longest mapper runs are around 560 megabases. Read length is easy. We have done no read length research for a long time because we just know that the pore will process pretty much what you give it. And at 40K, here's a 50 KB fragment, at 40K, the signal looks very like 10K. So there's no loss over time, as you often get with other systems. Which again, for a long read system, is just perfect. Now I'll zoom on a bit now, because these are now different pores. It turns out there are lots of pores that work to varying degrees, and they can be engineered and tweaked to give better signal. Now we have a pipeline of pores coming, and the next major upgrade or change is a pore called R8. And the main difference with R8 is that it has a completely different squiggle shape from R7. So all of its errors are different. And I would anticipate that will be available on its own. Actually, it's more GC friendly, and the 2D data will be better. So either that can be used on its own, or it can be used in combination with R7. And again, I'm predicting, I'm not stating, that things like SNP calling will be significantly boosted by using a 2-pore system. Just to emphasize, we do not use lipids, we use a custom polymer of our own development. And again, it's key. Now the MAP, I've got a couple of minutes left. The MAP program has been going on now since early June. Again, Oxford Nanopore is a very unconventional company. This is a sort of very open but not completely open access program. MinION is clearly not designed for genome centres and core facilities. It's designed for individual PIs who want to do everything from experimental design to sample prep to analysis. And so most of the people who are on MAP are like that, and we had several thousand labs applying for over 20,000 MinIONs, and we've let in around 500 labs, I think. I don't know how many MinIONs are out there. Now, over time, for people who are waiting to come into MAP, I anticipate there will be additional inclusions over time. So flow cells kits are shipped, and there's an online support portal. And shipping from day one went around the globe. I think it's 20 countries. It could be more than that. We had some difficulties getting things into New Zealand and other places, but I think that's now sorted. Shipping causes a considerable number of issues, and again, part of our objective was to sort those out. We generate several hundred flow cells per day, and it's thousands or tens of thousands per month. It's easily enough to support and scale MAP going forwards. The system is designed to be highly manufacturable. Okay, I'll zip ahead a bit because this is somebody tweeted I'm boring them. So, little look in the skunkworks. So there's a new MinION coming. This is really MinION 1. What's out there is really sort of beta, and this has all the fixes and tweaks in it and other things. A much neater little system. You'll notice there's a slightly different looking flow cell here. We're also diversifying the MinION into a family. There will be other versions that are targeted at non-traditional markets, like, for example, cell quantification. Coming along now, and I think we're talking about this in public a bit more, we have a new product coming through next called Promethion. And it turns out that we can take the innards of a MinION, the core, and we can make a multi-core system, such that we can have a very large number of channels operating on a very small sample volume. And those samples, because of some new technology, can be loaded from the top, which means it's accessible to pipetting or dispensing or other laboratory automation procedures, such that one human genome might be running on 96 wells, or you might have 96 microbiomes running on 96 wells. It's completely down to the user. We're going to showcase these at ASHG 2012. One thing I will tell you is it's pavestone heavy. It's not something you want to throw at your postdoc. Now Promethion, again, the number of channels is going to be up to 100,000, but that puts you into a very high throughput range. Even at current enzyme speeds, you're looking at maybe 300-400 gigabases per day. And with long run times, it should be a multi-terabase machine. If we put more channels in or speed up the chemistry, I think it can be over a terabase a day. And that is coming along much sooner than people probably anticipate. Another key area of focus we're going to circle back to is sensing directly from biology. I'm very keen on this. To put the thing truly in the field, you can't have a lab. You have to just touch something to a sample and then be able to generate sequence data. So we're enabling that. And the first version of that is a system we call Spot-on-T, which looks like this. It's a modified flow cell, and that transpososome system that will be encapsulated in some form, such that you literally put sample on the consumable and it does everything for you. And in this case, what we do is we envisage possibly having a matrix in here with the one-step transposase on it. You put sample on, it processes that sample, and it diffuses through onto the pore array. I'm very keen on that. Something else that's in the skunk works is direct RNA. People are quite keen on this. Again, we just haven't had the bandwidth until we raise the money to do this. So we have proof of concept now on reading RNA molecules directly, and that is binding an enzyme and generating sequence-specific squiggles from those molecules. So I would imagine in the near future there will be a direct RNA kit. And finally, if it's let me, I'm 1 minute over. It is very difficult to talk about this because it's basically software and concept. But a significant amount of effort is now being put into this thing called Metricore. And it's a cloud-based service, nothing particularly interesting about that. But it exploits the streaming nature of the platform. It means you can fire off DNA molecules like tweets to this thing, and it will process them and tell you about them. Now, where we see that going is in non-traditional areas, so out of laboratory, for example, self-quant, food production line, biosecurity, areas where DNA sequencing you don't want to have lab. The way that works is, I think Metricore will effectively sell a software service with vertically integrated devices. If you think about your jawbone up, your Nike Fuel Band, something like that. And of course, if we're accumulating context and locale specific DNA information over time, that becomes a very powerful resource for, yes, big data, which I love, by the way. And so I think that the Internet of Things can be tracked and quantified using live streaming DNA from very cheap portable devices, and that's where Metricore is going. Finish up. So nanopore sequencing is here after 20 years. I know we have haters and we have lovers, and in the middle is a huge swathe of silent majority who are just waiting to see how this pans out. The first practical nanopore device is MinION. It has been manufactured, shipped and sold at scale since June, and we're hammering out those bugs. There are field bugs, but I'm pretty confident we're making progress there. And I think, you may not believe this, you may be comfortable with big machines and high throughput and all that stuff, but my prediction is that this device or something like it, this technology, maybe us, maybe it's somebody else, will change everything again. That's my view. So with that, I will stop and see if I can stop screen sharing, and there we go. That's the end of the talk. Over to you.
Brilliant. Thank you very much, Clive. That was awesome. I have said on Twitter we'll take some questions. I've got a couple, only a couple, so you must have addressed most of people's problems. But Nick Shirk is asking, is there any news on progress reading proteins?
Okay, so in the background at Nanopore and with our academic collaborators, there is a lot of progress on proteins. I don't have to talk about it, and frankly, contrary to what's been written up recently, a lot of it has to stay secret because people are still filing patents and all the rest of it. We will do proteins. There will be a protein analyzer version of this platform, and when it's ready, we will talk about it.
Okay, great. Another question is Craig Duffy. He's asking, how well does the MinION handle extremely 80 rich or poor genomes?
Now, we've had some early feedback from that. Hasn't been good in field, but we know what's causing that, and I think it's going to get fixed. It's largely a software problem, and I'm hoping in the next week or so, people who've been doing extreme genomes should see that getting fixed up. More so if they do 2D. 2D naturally fixes that problem. It's a base caller problem. It doesn't know you're doing extreme genomes, and it's parameterized in a way that it biases the wrongness or something like that. It turns out if you get high 2D yields, it self-corrects. But if we also reparameterize the base caller, it will correct. So my advice to those people who've been doing that would be to please try and get high yielding 2D runs on those samples.
Okay, and I mean, a more general question. What do you think is the opportunity for bioinformaticians who are interested in contributing, particularly to base calling and difficult problems, actually contributing, especially if they're not part of the Access program at this point?
Okay, so we are constrained by informatics. It is quite hard to hire very good people. It does limit the rate at which we can implement features and fixes. That's true. MAP is fairly open, but has a moat around it. We made all the files and all the other stuff open, and we tell everybody how it all works. And you can go in there and do what you like, and we sort of try to help people on MAP with hints and tips. That slide was from an update that told people about unmodeled data that we haven't got to grips with yet. So I think there are informaticians on MAP who are digging in there. Now, beyond that, I think if MAP has a self-certification process where if you're happy with what you're getting, you can self-certify rather than the company doing it, which is again very unconventional. You can self-certify and then you can start doing what you like with your own data. So I know that you're about to release something and other people are releasing things. So I think if there are people not in MAP, that's a good route for them. We didn't let pure bioinformatics people into MAP because it's not aimed, MinION isn't aimed at bioinformatics people. It's aimed at PIs who want to do everything. So almost all the successful mappers are people who want to run the machine and analyze the data.
Yeah. Okay. And one from Phil Ashton. So on that kind of theme, have you thought of any alternative weird schemes that might base call better than hidden Markov models?
You know what? I annoy my informatics team every week by thinking of those things. The point where they tell me to shut up, basically. I think, as Li Heng said, algorithms don't just, it takes time and thought to get it right and to test it. And then you've got the parameterization. And then you've got to deploy it. And then people do things you don't expect. I'm sure there will be other schemes that are comparable.
Okay, and this sort of ties into a question from Nicola Rubin. So if you have the FAST5 files, is that going to be enough raw data to be able to implement a base caller from the kind of raw squiggle data?
No, so what the machine does is it writes out this enormous blob file. As you can imagine, if you're sensing at 5 or 10 kilohertz per channel on 500 channels, you get a lot of binary data. And then they split out from that the HDF files that contain the squiggle. And then when that gets sent off to Metricore, it comes back with the base calls. So from the HDFs, FAST5s, you can work on squiggles, assuming that the squiggles are correct, that the step before that got them right. But the machine, if you're running a machine, that's why it's so important, the raw data is all there. You can go into the raw data, and there is a script circulating around that lets you do your own extraction process. So you can go right the way back to raw data, a bit like going back to images on this Alexa system. Okay. I know that there's somebody out there I know who is doing raw data only analysis, for example.
And are those files much bigger than the FAST5 files, or
enormous?
A bit bigger? Enormous.
Enormous.
Okay, so you have to capture those specifically when you run the instrument.
They're written onto the hard drive of the laptop.
Yeah. Great. Okay. Well, I think in the interest of keeping to time, I'm gonna carry on and go to the next talk, which is me. So that's straightforward. It's worth saying that this is me, in case you haven't met me before. We've got 262 viewers online, which is way more than the 100 or so that registered. So it's very nice to have you all here, and lots of nice feedback on Twitter. So without any further ado, I'll go into my talk. So I'm going to talk about nanopore sequencing. I'm going to talk a little bit about bioinformatics opportunities and challenges. So the first introduction, the first thing to say is this is a tricorder made famous by Star Trek, but very much now adopted into the sort of mindset of people thinking about the future, particularly of medical applications. And if you're a Trekkie, you know that there are various different tricorders, like an engineering tricorder that you could take down to a planet and understand its mineral composition and if there was an immediate threat to life. And of course, the medical tricorder would give you an instant readout, an instant diagnosis of anything, including diseases not yet really understood. And I guess the question is, and this is a question that other people are asking, is what might a real-life tricorder look like? Probably doesn't look like this. You know, what would it detect? And you can see where this is going in the context of nanopore, because my feeling is very much that a DNA-based assay, or a cDNA, or perhaps an RNA-based assay, is going to be very, very valuable in terms of giving us actionable medical information in a reasonable time scale. But you know, others may disagree. Others may think that proteins are the most important thing to look at. That's not really my particular interest. And there are other things we could detect in a sample. You know, look at its appearance on a microscope. This idea of a molecular nose that can detect metabolites, and there has been evidence that dogs can sniff out cancers, and biofilms produced by Staph aureus have a particular odor. But I'm very much of the belief that our first version of a real-life tricorder is going to be a genome-based assay, and there's already good proof of principle that genome-based diagnostics can do plenty of things that are relevant now. But Vogelstein and co. Have been pushing this idea of the liquid biopsy, this idea that we could take patient serum, sequence it, allow us to detect cancers by way of mutations that are in cancer DNA floating in the blood freely, allow us to monitor treatment, to see what happens when a patient has a colectomy. Those fragments go down, and then the patient perhaps has remission, goes back up again, and has chemotherapy and goes down again. And this idea that we could have a personalized assay for cancer and track how well treatment is working is a pretty cool idea. We know for things like IVF, the idea of doing pre-implantation genetic testing is important in terms of the success rate of those procedures. And we already know that sequencing can give us good perinatal genetic screening by exploiting the small fraction of free fetal DNA, and that's very sensitive for picking up Down syndrome. Clive already mentioned this idea of self-quantification. You know, what if we could just repeatedly take samples of our blood and relate that to our lifestyle, relate that to other symptoms we had? Would that tell us something intrinsic about health and disease that we don't yet know? And you know, for me, the most important potential applications, what I work on, is infectious disease and the opportunities here. So we already know in microbiology, and I'm particularly talking about bacteriology, that genomics can do an awful lot, whether we're looking at a genome of an individual isolate up to a population of microbes with perhaps some heterogeneity, right the way up to a metagenome. And thanks, Lauren Cowley, who I think is watching for that picture of a virtual metagenome. And we know that, you know, if we get genome sequence information, we can quite easily identify a species and a strain from that data. We can make certain predictions about virulence. Does it have a particular tox? Does for example, an E. coli have a particular toxin encoded, a particular phage sequence, or not? We can make quite good predictions, certainly for some species, about the antibiotic sensitivity. So, for example, we can very clearly work out the antibiotic resistance pattern of Staph aureus or TB just from the genome sequence. And that's kind of moving into the population because there may be heterogeneity there. And there are countless examples now of using whole genome sequencing for typing and tracking of microorganisms, both in terms of looking at local outbreaks in hospitals or in the community, or going right up the scale to sampling isolates from all over the world and working out the natural history of that particular pathogen. And this idea that we could use metagenomics to discover new pathogens is reasonably well established in virology, increasingly established in bacteriology, and it ties into the idea of the microbiome and how community dysbioses are important in certain conditions. Certainly we know it's important if you have a C. diff infection. That's essentially a dysbiosis of your gut microbiome, and that's correctable by these techniques such as fecal transplants. So that's enough of the kind of preamble. Talk about nanopore sequencing, and that's the kind of the view of why I'm interested in this. So I put this slide up when I spoke in Oxford last week, and this is, if you don't recognise this is a fanboy picture from Wikipedia. It's a particular type of fanboy who likes Nintendo stuff, wearing all of the gear there, some cartridges, some of Nintendo's less popular add-ons. And I guess I am a bit of a nanopore fanboy in the sense that I can see its potential. And I'm very excited about it, and I'm one of the people lucky enough to be in the MinION Access program, and so yes, I think I have some amount of natural bias as associated with that position. So my role is to try and get you enthusiastic as well, because ideally I want to be on the right side of history, and I want you to be as well. So what do I think is cool about nanopore? I think there are things that are demonstrably and objectively cool about it. One is that it's extremely portable. It's less than the size of an iPhone. It plugs into your laptop, so it draws its power from the laptop. That opens up possibilities, perhaps not possibilities we've realized just yet, but it does mean that you can take your sequencer to the sample. And that's obviously important for medical applications. We think it's likely to be quite cheap, although we don't know what the current pricing is. It's certainly very cheap for us at the moment, because on the MinION Access program we pay a $1,000 refundable deposit plus a shipping charge, and because we're through this process, we've received lots of flow cells, and so the actual cost of sequencing is pretty low. Whether that will translate into the commercial decision making, I really hope it will, because I think one of the opportunities here is to do lots and lots of sequencing, lots of small assays, and certainly that's what we'd need if we were going to use it for a diagnostic test. It's also objectively real time. The reads come off in real time. I can show you that, and you can analyze them in real time. So the reads get sent up to the cloud system called Metricore, the raw event data, and that's translated into base calls, and there's no reason you can't use that. Actually Matt Luce, who's going to talk after me, is going to give it a much more visceral demonstration of that, I hope. And clearly the reads have been promised to be long, and they are long. Now I should just say, for balance, that there are examples of portable sequencing already. So this is a fun paper from Rob Edwards' group, published in PeerJ, where he's actually taken an iron torrent PGM and all of the stuff you need to run it to prepare your library onto a boat, and he's driven it around the South Pacific Ocean, looking at the Southern Line Islands and looking at the microbial ecology. And the reason that he gives for that in the paper is that they can look at the results of sequencing genomes and sequencing microbiomes, and they can act on those results, and it kind of can inform how they do their sampling and what experiments they're going to do next. I tend to think it's probably just because they wanted to put an iron torrent on a boat. So portable sequencing, if you can call that portable, it is already a thing. But I would argue that this is a much better example of portable sequencing. This is actually this is genuine. I did take a MinION home, and I have there a nice cocktail I was drinking, and I was doing some sequencing on my Mac, which is running Windows over Boot Camp. Did some sequencing at home, generated some data, and I've also taken the nanopore down to a course that I was helping out with in Italy, and we got the students to load it and generated some data there. So I think clearly it is portable. Is it scalable? I think here's our version of scaling. Clive's talked about the PromethION. At the moment we have something like 5 or 6 MinIONs, but we've only got 3 laptops, and so our way of scaling is to buy more laptops, and we have had all of these running at the same time, and so that's one way of scaling, probably not the future way of scaling. Okay, so I promise you some data, and last week I did release some data, and I released it, gave a URL to people at the conference to download. That was coming off my server, but I will open up some data. So I just want to talk a little bit about how the library is created just before I talk about the data, because it's kind of important to understand that. So Clive kind of mentioned this already, but it is important that if you're going from genomic DNA, it does still need to be shared. So ideally you want to start with very high molecular weight DNA, and you want to do the kind of minimal amount of sharing to try and preserve long fragments. So we use the G-tube protocol, which is recommended, with a slightly reduced RPM speed of 5,000, and we're trying to encourage longer fragments in the mixture. And it is quite important when you're doing the sample preparation that you're going to need to be taking care when pipetting, trying to use wide bore pipette tips, and trying to disrupt the sample as little as possible, so that the long fragments persist through in the mixture, because they are quite fragile when you're talking about very long reads. It is important, as Clive said already, that you need to ligate several components onto the DNA. One important component is the hairpin. If you don't have a hairpin, then you can't generate complementary reads from after the template. And it's very important, obviously, to have the cholesterol tether and the motor protein, which will slow down the read. And there is a second enzyme or kind of structural protein, I'm not sure which, that helps slow down the complementary strand. And so getting all of those bits and bobs on the DNA are kind of vital for getting good data. So I did put data out actually ages ago now in June. I put one read out, which apparently is not enough according to some members of the community, but there is one read out there, and it was an important read actually, because it was the first run we did back in June after we'd certified on Lambda, which is the first step of the MAP program. And the first read we got, which was mapping and was interesting, was from one of our pet organisms, Pseudomonas aeruginosa. And this read blasted, reasonably well, and actually turned out to be a serotype defining read. So contained within that read is enough information to let you know which serotype of Pseudomonas you're dealing with. And it's kind of a small thing, but it's kind of super cool as well, and made us start thinking about how this is going to be genuinely useful. Okay, so data. So we've had kind of successful runs on the MinION, I think about 20 times now. We're on to about our 20th flow cell. These are some of the newer flow cells that we've run with R7 chemistry. And this data has been available for a little while, if I've sent you the link, but that was coming off my server in Birmingham, and I didn't want to kill it. And so we've actually put this data up onto GigaDB with the help of Scott Edmonds from GigaScience, and that's a citable DOI. You can do anything you like with it. You can publish on it. You can publish any analysis on it. We don't really care. It'd be nice if you cited us. That's available to download now. So there are actually 3 runs available, with the raw, not the raw data, but the intermediate raw data, the FAST5 files, which have those squiggles in. And so we have one from all from E. coli, one R7 run, which was done with a protocol which doesn't have an overnight incubation, and I'll explain that. 2 runs from E. coli where we do have an overnight incubation, and the plan is to add 1 more run to this data set very soon. And the reason it's not up there yet is that it's still actually running, and I might try and, I won't do this now in case I break something, but I'll try and show you that in a second. So the latest run is running with the latest chemistry, R7.3. So I have analysed a partial run just to get a feel for what that's doing. Okay, so if you download all these files, you're going to need to be able to look at them, and we obviously recommend you use our toolkit, which is called Portals. So this is yet another example of where Twitter kind of comes to the rescue, because when I was getting my reads initially, there's absolutely no scripts at all supplied by Oxford Nanopore to even get the most basic analysis done, getting fast A or fast Q out at the time. So myself and others, including Keith Roberson and Mick Watson, started writing toolkits, and I started putting some stuff on GitHub. And Aaron Quinlan quite luckily got in touch very early on and said, "Oh, I'm also building some tools. Shall we team up? Shall we combine forces?" And that's lucky really for me, because Aaron is a much better programmer, much better at building software and packaging software. So he took some of the stuff that I did and some of the stuff he'd done, put it together in this toolkit called Portals, which is now published in Bioinformatics and Application Note, and we're continuing to add to. So you need portals to actually get data out of this data set, although I have extracted the FastA files for you as one of the entries in the GigaDB. So this is the yield plot from a non-overnight incubated library from the R7 chemistry, and this just gives you a feel for how many reads are generated in a kind of sort of typical run. This is not by any means our best run, but it's probably above average. And what you can see, we've got time plotted on the x-axis, reads up the y-axis. You get a decent number of reads within a short amount of time. So we already have 10,000 reads in around 6 hours. But you can just keep running this thing. There's no reason you can't just keep running it. But you can see that the performance tends to tail off around 72 hours. We don't think there's much advantage running it more than 72 hours, but the standard protocol is 48 hours. So I guess it's probably important to think about both yield but also throughput, because typically you're not going to run this for 72 hours on a single sample. You're probably going to run it for a certain amount of time and then maybe introduce another sample. And what you can see from the run stats is that the number of forward reads is about 43,000, number of reverse reads about 23,000, and so the 2D reads is always going to be less than the number of complement reads you have, because you need template and complement to make a 2D read. And what you can see here is that most of these reads are categorized as normal, in quotes, and few of them are categorized as full, and I'll explain what that means in a minute. So in terms of is this a typical throughput, these are the run metrics. There's a competition on the MAP forum for people on the program, and if you win this competition, then you get sent some more flow cells. So you can see that if you are early winners like us, you are at a slight advantage, because you'll keep getting flow cells sent through. And you can see that our group, and it's my name there, but it's really Josh Quick and Simon Callis, who have been responsible for running the instrument. Our group has been responsible for 3 of the winning weeks, and actually also the highest throughput record for a mapper in week 4. We can plot the data in various ways using portals. So, for example, you can get a pore occupancy plot, and that's basically showing you for the array of 512 pores on the flow cell, which pores are generating information and which aren't. And in this case, you can see that a smallish number of pores actually are responsible for generating most of the data, and some pores are generating very few reads, and that ties into this idea of a duty cycle that Clive was talking about. So this is a pretty average, not particularly great flow cell, which QC'd at around 250 pores. So read length is very long, and from data I'm not showing but I've seen, it does seem to map on very nicely to Bioanalyzer, although Bioanalyzer has a slightly different way of calculating, I think based on either mass or number, I can't remember. So you do see a slight difference, but generally speaking, if you put long fragments in, you get long fragments out of the sequencer. And you can see here we've got a mean read length in this run of about 6 KB, and quite a long tail of long reads stretching out to over 45 KB. I don't show the data here, but you can obviously check this for yourself. But read quality is really not a function of the position in the read. It doesn't tail off in the way that you would expect it with other sequencing. And conservatively, at least 80% of these 2D reads map with last said, and the mappings are good and long. Okay, so if we take the set of 2D reads, and really what we've focused on is 2D reads, we've tended to ignore the forward and reverse reads in favor of 2D reads just because they're higher quality. And we can see that taking 2D reads here, we can cover the E. coli genome around 10x coverage, and you can see a nice Gaussian distribution of coverage. And in fact, in this run, only 15 bases in the entire reference genome remain uncovered. So we've essentially sequenced the entire genome. Wouldn't surprise me if those other reference bases were not actually in the genome. And this slide really is showing how things have changed with these chemistries. So what we want is these reads that are called full 2D, and full 2D is a read where you have both template and complement, but critically the complement strand is slowed down as it goes through the pore, just like the template strand is slowed down. If you have full 2D, then you have better quality. So what this slide is showing is actually what happened is originally the protocol didn't have a step called overnight incubation, and what you can see the effect of that is that the proportion of full 2D reads is very low relative to the total read data set. And so the overnight incubation was kind of brought in, I think it's fair to say, as a bit of a kludge to try and increase that fraction of full 2D reads, and which it does, but it also has the effect of reducing the total yield. So what's happened in the last week or two is a new chemistry has been released, a new genomic DNA sequencing kit called R7.3, and that's what we're running at the moment. And that seems to have had a decent impact in the both the yield, so it's doing decent yields, like we were seeing with the non-overnight incubations, but you're also seeing a much higher relative proportion of full 2D reads, which is good. So this is what I'm talking about in terms of, so this is the key kind of quality slide, if you like, for E. coli. This is if you don't have an overnight incubation, and what you can see is that the forward reads are slightly higher quality than the reverse reads, and that relates to that slowing down through the pore. And it also means that really there isn't much difference between normal 2D and full D in that case. If you do the overnight incubation to increase the number of full 2D reads, you see this kind of distribution. So you see that the reverse reads have improved in quality because they've been slowed down, and there are more of these full 2D reads, and the kind of raw read accuracy. And this is by doing 1 minus none mismatches over a line len with last is it's just a bit under 80%. So what can you do with this kind of data? Can you call SNPs? Well, yes, we found you can call SNPs quite well. So this is just IGV showing, I think from Salmonella actually, not E. coli, but this is showing a SNP in Illumina data, and a SNP in nanopore data. Obviously the nanopore alignment looks a bit messier, but you can convince yourself that there is a SNP there by setting an appropriate allele frequency. And if you plot allele frequencies, this is back on E. coli and 2D reads, what we can see is that the mean allele frequency is about 12%, so just over 1 in 10 bases at any given position in the reference is incorrect, and the rest is the correct allele. And so you can see it's going to be a bit tricky to call hets with low coverage, but in bacterial SNP calling, if you set an allele frequency around 50–60%, you can generally partition true and false SNPs. Okay, so in conclusion, nearly the end, I mean, I think there are some quick bioinformatics wins up for grabs, and this was a slide I presented in Oxford, and I suggested that it would be quite straightforward to scaffold a draft genome with this data. I hope it would be possible to do a de novo assembly, either through an Illumina correction stage first, and then an overlapping assembly, or perhaps, a la HGAP, doing a pure nanopore de novo assembly might be possible. And in fact, I saw that Mike Schatz had tweeted when I woke up this morning that they'd done a overlap-based de novo assembly using Illumina correction, I think. When you look at these alignments, they're a bit messy, and they quite clearly need some realigning, and ideally that would take into account the error model. And I think we need a consensus caller that is a little bit more KMA context aware, or even ideally signal aware, and I guess this is something I would love to see the community kind of take up. So I have agreed with Scott Edmonds from GigaScience that we will try and build a continuously integrating server for this data set, such that if people want to contribute recipes, either alignment scoring matrices or aligner parameters or other such things, then we can incorporate that and make it continually integrating manuscript based on this data, which everyone can be an author on if they contribute. So I was going to finish there, but I had a very exciting email in my inbox earlier today, which I wanted to share. So just check whether you can see these other slides. Yeah. So Anton Korobenikov, I can't say that. I shared this data set with Anton Korobenikov, who is the author of Spades, a very well-known and excellent de novo assembler, which will handle Illumina and PacBio type of data. And he wrote to me and said that he unfortunately can't present at the meeting, but he wanted to. He wanted to present. Wanted me to present these slides. So thank you for Anton for sending these over. So Anton's taken this data set that I've released and he's put it through Spades 3.2, which is his latest development version. Although he thinks similar results should be obtained with the 3.1 release, which is available. And he's taken an Illumina data set of K12 MG1655, corrected it in the usual way with Bayes hammer, and did initial contig graph construction with the Illumina data, and he's incorporated these nanopore data sets for repeat resolution, scaffolding, and gap closure. And so he's actually reused the read to graph aligner that was developed initially for PacBio reads to align the nanopore data to the Brown graph that the Illumina data produces. So what are the results? Well, the results are pretty damn impressive, I think. So with the Illumina-only data, he's getting around 92 contigs, largest contig 285 kb, with an NG50 of 133 kb, about 98% of the genome covered. And what he's found, if you add in the nanopore data, you get 4 contigs greater than 500 kb. And in fact, that's a bit misleading, because the largest contig is the same as the N50, and covers the entire E. coli reference, and so essentially he's produced a single scaffold assembly with this nanopore data, and he's done that in a matter of days without really doing a huge amount of work, I'm assuming. He does detect 6 misassemblies in this assembly, but when he's dug in and looked at those, they are associated with transposon repeat units, and when he compares this against a PacBio and Illumina assembly, it's the same 6 misassemblies, and so this almost certainly represents a difference between the K12 MG1655 that we've sequenced and the one that's sequenced by Illumina. So we'll generate some Illumina data just to confirm that as well. So essentially, single scaffold genome, fully covered, at 4.64 megabases. So he suggested there's a need for a few improvements. Perhaps the read to graph aligners could be improved for nanopore data, and it's unclear how much nanopore coverage if we want to do hybrid assemblies where the nanopore data is used for contigging. So that's his acknowledgement slide. So I'm very, very grateful to Anton for sending that through. And I'll just go back to my slides. Aware that I'm running over a little bit and put my acknowledgement slides up. So just particular thanks to Josh and Simon in my group for running the Nanopore, to Aaron, and to all the chaps and chapesses at Oxford Nanopore for their assistance in this program. Right, so it's back to me. So I've got quite a few questions, I think. Let's try and get my face up so I can react to the horror. Here I am. Okay, this is really hard work. Okay, so Marina asks, how is the accuracy of 2D reads compared to the accuracy of PacBio CLR reads? Don't know really. I'm sure someone else can answer that on Twitter. So Aaron asks, can you elaborate how you distinguish full from normal 2D reads? Is it complement events more than template events? Yes, Aaron, that's how we do it. So if you see more complement events, we assume that the strand has been slowed down, and they're called full 2D. And Portals has got a method for pulling just those reads out, if that's what you want. Ryan Johnson says, do you think this could be the start of the end of our reliance on dedicated sequencing centres? A bit controversial. I think probably my feelings on that are quite obvious. But you know, we're still going to need Illumina data for a time. Right, I can't see any obvious questions that I want to answer. So good, all right, brilliant. So I'm a bit over time. Does anyone on the Hangout wanted to respond at all to anything I've said? Clive?
I will just looking at your numbers there, Nick. You're still lagging behind us by quite a way.
Okay.
About half of what we're getting. So I think there's still some room for us to help you there.
Yeah, I think. Yeah, so that's good.
But obviously a lot better than it was, clearly. Yeah,
yeah. And I think importantly the data are quite usable. So that's the most important thing for us. I mean the experiments that we can do now with say 80% accuracy are very similar to the ones we could do with 85% accuracy, and actually some quite interesting stuff that we can do. And Josh is going to talk about that.
Right.
Anything else? Okay, so all seems to be going very well, assuming everyone's still out there watching. And so it's my great pleasure to hand over to Matt Luce from Nottingham. And Matt's going to tell us about some software he's developed for real-time streaming of Nanopore. So over to you, Matt.
Thank you. Great. So yeah, hi, my name is Matt Luce from Nottingham, and it's good to have the opportunity to talk. So today I'm going to be talking to you about some work we've been doing to try and develop real-time analysis of MinION data. And I just wanted to give you a little bit of context at the start as to what we're doing on the program and why we're here. So part of my job here is to look after DeepSeq, which is the University of Nottingham's sequencing centre, and we actually actively encouraged people to apply to the MinION Access program here in Nottingham. We wanted to get some exposure to the technology, find out what it was like. And so we're currently sort of helping support several people with different projects, and as Clive alluded to, these are, how can I delicately put it, typical bench scientists. These aren't bioinformaticians. I'm not going to be talking about anybody's data and really exploring it in that much depth. I want to show some of the real-time things you can do, which are quite cool. So obviously, first day with a MinION, you know, you do your library prep, it's great. You do your loading, you run Metricore, which Clive talked about some of the things that's going to be able to do as well. And then you get back these HDF5, FAST5 files, and when you're sitting with those with somebody, you kind of wonder what you're going to do next. Where do you go with these? And the first time we ran it, we were there, and there were some users looking at that, and as soon as the data comes back, there are really obvious questions you want to ask. Do my reads map to the data, to the reference? Is my library prep any good? What does a good library prep mean? We've learned a bit about that. What's the coverage? Should we throw on more library, longest read, all these things. And as I say, we had several people on the program, and we wanted to look at the data straight away. So I remember on that first day very well, we downloaded HDF View. If you've not used that, it's great for looking at HDF5 files. We got the FASTA sequence out. We ran BLAST in that case. Not the best aligner, it turns out. And we just went around in a loop looking at reads as they were generated. And it really is quite exciting to see this data coming back in real time. But the obvious sort of light bulb moment is, well, we don't want to sit here doing this. We want to do it automatically. And so we've done a little bit of work to try and do this. So the MinION workflow, as Clive's told you, here's Sunir. He's working in the lab. This was yesterday, actually, working with an R7 chip, which is still running today, and I was hoping to show you some data. I don't know if we'll get there or not. But the raw trace files get uploaded to Metricore or Biometricore. They're base called, and then you get back the HDF5 files, FAST5 files that you really wanted to look at. And we wanted to ask the obvious question, can we capture that return data and store it? Can we do something with it? And initially we were, this was before all the tools that Nick's been alluding to were available. So POR tools from Nick, I know there's an R package POR from Mick Watson as well. But we wanted to try and get these reads and we wanted to process them. But we didn't want to interrupt the run on the machine controlling the MinION. So initially we were copying reads off at the end of a run. You've got to wait some time. You want to know the answers quicker than that. So what we did was mount the folder that the reads are returned to on the Windows machine on a Unix box over a local share. And then, of course, we could watch the data appearing in that folder. We could watch the reads coming in. So we wanted to catch the read, we wanted to align the read, we wanted to store the data, and ideally we wanted people to be able to look at what was coming back in real time. And so we've got some a Python script that runs at the back end that sits there and it watches what's happening. It collects the data. It will configure a back-end database specific for each run. At the moment, we're working on different ways of doing that. Stores the data, takes everything out of the HDF5 file, puts it into an SQL database. It aligns to last, and then it goes round and round and does that until a run is complete. You have to tell it information about the reference you're aligning to. And at the moment, if you switch a run, so if you stop your MinION and you change it to something new, and you want to align to the same reference, you can do that and it handles it. The only problem is if you're changing to a different reference, you have to stop the script and start it. And then we've built a really basic website at the moment at the front end just to look at that data. So what I'm going to try and do, obviously, sorry, before I give you a live demo, obviously there's lots of benefits to having this information in the database in terms of visualizing what you're looking at. You can archive data, you can share data, but the amount of data that's being generated is rapidly becoming a problem. We're getting so much data off the flow cells now that we can't process it quick enough at times. So the current model for this software for people who want to use it elsewhere is to do a local installation with one of your MinIONs. We're not thinking about a central repository, and obviously Clive was talking about Metricore maybe doing these things in the future. For the purposes of the demo that I'm about to attempt, and this will be interesting, a live streaming demo of some software. I'm showing some runs that are early runs. They're R6 runs. They're before we knew what we were doing, perhaps at times. We've made some mistakes. We're getting better, and I was hoping to show you the R7 data, and I'll explain a little bit later why we can't do that. So hopefully you can now see a view of our very basic web interface. I don't know if you can see the mouse on screen or not, but you've got various options here. An overview tells us how many runs are in this database currently. There's 3 runs available. There's a current sequencing run, which is actually us uploading some of that data from Nick's that Nick has made available. I'm not going to show you that today. You can also obviously look at previous runs, so there are some examples here of previous runs which we're doing. Now this is amplicon sequencing, so you can get a summary, and don't pay too much attention to the numbers here because, as I say, we know we were making a mistake, and I'll explain in a moment how we know that's very true, but graphs being generated here, the number of reads called for the template, the complement, the 2D, the average length, the maximum read lengths, coverage, all those things. And the thing that told us we were making a mistake down here in the run summary, they had 8,000, nearly 9,000 reads generated from this particular run. Very few actually resulted in sequence. And it turns out this was a complex amplicon, it was a fusion PCR, and there were some problems with that. But you can browse around, you can look at this data, you can look at the rate of base calling over time, read lengths over time, all these sorts of things. We also plot the pore activity. So I'll show you this is quite fun. The number of active channels. This then tells us what was happening here. We've got the number of reads per pore in this heat map here. And this is the number of reads that result in sequence. So the FAST5 files that were returned contain sequence data. And then there's a number here below, which is the number of traces. This is the number of FAST5 files that were returned per pore. And clearly there's an issue. And a lot of these were very, very short runs. Very, very short sequences. There was a problem. So there's a lot of things that we can do there and look at. You can also drill down into individual read data if you want. Note at the top of the page here it says under development and features won't work, but that's because it's a work in progress. Let's pull up a single read here. This will give us a quality plot for that read, or should give us a quality plot for that read. There we go. It's filling in the data. It gives us some of the information on on the parameters of the run at that moment in time. That's why these HDF5 files are great, because you can drill down and get some information from them. And you could also export FastA or FastQ. So this is a quick way of finding your longest read and seeing what it is. You can also export the reads FastA, FastQ format, the reads that align or the reads that don't align, and so on. So what I'm going to do now is attempt kind of a live demo of what it looks like during a run. And just to be clear about what we're doing here, this isn't attached to a MinION in real time, but what we're doing is copying files from one folder to another at the same rate as they were generated by the sequencer in the first place. So it's as close to a real run as I could get, and hopefully it's going to work. So that should be running in the background now. I'll just see if the scripts are picking things up. It should start to pick things up now. As ever, don't work with animals, young children, or live computer demonstrations. Okay, are we getting anything? Let's see. There we go. So you can see there's a message in the top right-hand corner there that's popped up. There's a run started. So we can go in and we can choose this run. We need to make sure we pick the right one. So here it is. And we can look at the live data. And what you can see here are the reads, the template, the complement, the 2D. You can see the numbers increasing, the average read lengths changing, the maximum read lengths coming up. With these two plots here, we've got percentage of reference with read. This is the percentage of the reference sequence that's covered by a read. It'll take a little bit of time for that number to catch up. And we have an average depth here of sequence positions. You can see some of these graphs being plotted out here for the rate of base calling over time. This is copying slightly faster than the run went. Read length over time, all sorts of things, number of active channels. You can see the reads popping into individual pores here as they're generated. We have quality summaries and various other things. So, you know, there's all sorts of things you might want to do here. I'm not quite sure why this percentage of reference with read isn't showing up, but one of the obvious things is you want to ask questions about the data and you want to know about it. So something we've been working on in the last couple of days is, you know, give me some information about when I hit a certain coverage depth. So let's say, you know, I want to know when I hit 15x on this run or 25x. I'll just set up a couple of alarms here. So we'll go back and look at the data as it comes in. So these views are available. The other thing that's quite nice, if we go to this data summary here, I'm not showing you yet here, but we can get coverage detail. So this is giving you a coverage plot of reads as they align to the reference in real time. So at the top here you can see this is again an amplicon. It's a short fragment that we're sequencing. And you can see the coverage there, and this updates as we go through. There are a couple of steps there, and we know in the background data. You can see about 1,500 bases here. We know from the experiment that's underlining this that there is some significance to that. So it's quite good to be able to look at that. We've also kind of plotted the 5' and 3' mapping positions of some of the reads. Again, some of this is a consequence of the actual experiment. But you can ask any questions that you wanted to of this data. You can also, as the run is running, you can export FASTA and FASTQ files. So if people are really impatient, they can start downloading their favorite sequences early on. At the moment, we're not talking about the alignment. We're not talking about aligned or unaligned reads. And you can see in the top right-hand corner here, we've just got an alert that you've got coverage exceeding 15x on the template strand. So that's good. So if that was all you wanted, you could run off now and wash your flow cell and start loading something else. And if we come back to the live data, you can see that's being reflected here. So it's kind of quite fun to look at the data this way. It's quite informative. You can pick up issues as they're happening. You can see whether you're getting alignments or not. You can see whether your library's okay. You can also send this to people who are using MinIONs and they stop coming and asking you questions, and you just see them sitting staring at a computer screen in real time, which is quite fun. And blasting various things off. The website that we've built has user management. You can load multiple runs into it. You can have more than one run being loaded in at one time. Having said all of that, we have recently tried the latest R7 chips, and you need a solid-state drive, you need a separate SQL machine and a web server in order to deliver that data in real time. And you can just see there's another alert there. We've now covered 25x for the template strand. So there's all sorts of fantastic things you could do with this. You could imagine screening for particular pathogens and being alerted if a sample gives you picks up a particular sample or species that you're worried about. Those alerts could very easily be even sent to your mobile phone rather than you having to sit and stare at the screen. So that's a pretty quick overview. Just see how we're doing for time here. So it's about time to wrap up. So I'll just jump to the last couple of slides. So where are we at with getting this out to people? We presented it last week in a poster form at the UK Genome Science meeting. It went down really well. A lot of people asked us if they could get hold of this. We're going to try and get it out. We've got a few browser issues. Those graphs, as you get more and more data, get slower and slower, particularly on Windows Explorer. So we're going to try and deal with that. We want to be certain about the processing requirements. As I have mentioned, the R7 chips are swamping our databases at the moment because of higher read numbers, more active pores, better read alignments. Everything just generates more data, which is great, but we need to be able to handle it. It would also be useful for people to be able to extract those original FAST5 files back from the database. We want to be able to do that. So for those who are after this, we're going to be trialing it in the next couple of weeks with a number of key sites, just people who won't mind if it goes wrong. And then we hope to make it available to all MAP participants in the near future. With a fairly easy-to-use system. We're thinking about remote, can we do this remotely for people? If we did that, obviously that would be short-term data storage. We wouldn't be keeping data for a long time. So just to kind of wrap up and thank the people involved. So at Nottingham, the DeepSeek team, it's Sunir Malla who's been doing the library work. Martin Blythe spent a lot of time working on some of the Python scripts. Got various MAP participants at the University of Nottingham who've been really helpful, and Nick, of course, who's provided some of that R7 data, which I've not had time to show you today. So if you want access or more info about it, email or tweet. There's also a link there to the poster that we showed last week at the UK Genome Science meeting. So feel free to have a look at that. So I guess that's me done. I should attempt to hand back to the live stream. There we go. That's me.
That's brilliant.
Hand back to Nick.
Excellent, Matt. That's really, really exciting. You sort of see where this is obviously going to go
Cool.
in terms of real-time analysis. Yeah, I guess my question is just when can we get our hands on it, really?
Well,
already.
Soon, I hope.
Yeah, and I mean, going forward, what do you think the role of cloud computing services and distributed services are in infrastructure like this?
Well, it's really interesting. When we first started doing it, what we'd really like to do is get this running on the same machine that's running the MinION. So have it sitting there locally, so you can just run the MinION and look at your data back. And for some of those tricorder type applications, that's what you want. You don't want to be having to stream data to different places. On the other hand, as the MinION technology scales up, as you get to the Prometheon type stuff, that's for Prometheon, isn't it? As you get towards that, it's going to be very difficult to do that on a local machine. So it'll be really interesting to look at cloud stuff there.
Yeah, and is it worth asking Clive, how this might tie in with any plans that there are at Oxford Nanopore?
Well, I guess if there's no point carrying on developing it because Metricore's going to do it all next week, that's great. But if not,
well, you're a little bit ahead of us, I have to say, and I think Metricore is aimed at specifically, really not, frankly, lab research and academic applications. It's really aimed at commercial users who right now don't do this. For example, food production lines, farmers, those kinds of people for whom they would have to buy a vertically integrated device that would just work with Metricore. There will always be the academic research and other more conventional domain within which we don't have ambitions to commercialize software. So unless you want to dominate the world and become a Google of streaming DNA,
it would be fun, wouldn't it?
Basically what I want to do, you're fine. If you want to do great academic, centralized bioinformatics services, I'm not going to take you on, basically.
All right, so you're safe for a few weeks anyway, Matt, sounds like.
Yeah, I'm sure other people will be doing it soon.
Okay, good. All right, so we're a bit over time, which is mainly my fault. So we'll move on without ado. Matt's on Twitter, I've just seen, didn't actually realise that, at Matt Luce, so you can ask him specific questions about the software. So I'm going to move over now to Josh Quick, who is a bioinformatician in my group, who's going to talk about some work we've been doing on Nanopore. So he's going to come and take my seat, which is interesting. So you want to introduce yourself, Josh, and then I'll start the slideshare.
Hello. So I'm Josh Quick. I'm a PhD student in Nick's lab, and I'm going to talk a bit about some applications that we've been working on. So contrary to what you might have heard this week, we've actually been working on some real-life applications, and I'm going to present some results now. So we've got some really loud drilling going on in the room.
We've done pretty well up to this point with technical issues, but someone has decided they're going to start drilling outside, so I'll just close the windows. That helps a little bit. You're just going to have to go through it.
Okay. So sorry about the background noise. So, oh, there it goes. So we're going to so at the time when we had just started the map, we'd been running some R6s, and fortunately for us, there was this salmonella outbreak in Birmingham. Fortunately for us, unfortunately for the patients, it's widely publicised in the press, and it was caused by Salmonella enteritidis. So we decided to go for it, and we thought we'd do some nanopore and some MySeq sequencing. The same day that we received the samples, the same day that we received the samples, we sequenced 16 of them on our MySeqs using a recipe called draft sequencing, which I wrote for the MySeq. Can sequence 16 bacterial strains such as Salmonella in 6 hours. So we received these samples at lunchtime. I did the library prep, and we ran the instrument overnight, and then we had the data analysed for an infection control meeting at 9 AM the next morning. So we were able, from that MySeq data, to understand that we did have an outbreak. So we had 14 identical strains. We had 2 which had 1 or 2 SNPs, and they were they were associated with in host evolution. So then we decided to take one outbreak strain from the ones we sequenced and one non-outbreak strain, and we sequenced them on both on R6 and R7 on the MinION. And this is just to show you that of all of the isolates that we sequenced, the outbreak was quite closely related to isolates that had been seen in Manchester, Thames Valley, and others in London. So we took the DNA, we put it through the standard Oxford nanopore library preparation, which you can see here outlined. It uses the G-tube with end repair, A-tailing, and ligation. It takes about 92 minutes. This week we've just taken delivery of the new sample prep kits, and that takes it down to 62 minutes by the loss of the HP motor incubation. It's now pre-bound to the hairpin. So from the two runs that we did, this is some plots showing the yield over time and the mean read lengths. So we're getting fragment lengths of around between 4 and 6 kilobases, and we've taken the output of the run and we've binned it into 10 minute chunks, so you can see the data being generated in real time. And so what we've done here is we've taken those chunks of data and we've mapped them to the Metaflan2 database of taxonomic defining taxon defining genes, and you can see over time that in the non-outbreak strain we were able to detect that it was a Salmonella enterica within 20 minutes. In the outbreak one, you can see within 10 minutes that we've got a Salmonella enterica. And really interesting, we weren't expecting to see this, but you can actually see two different chromosomally encoded phages showing up. In the non-outbreak, you've got the phage GIFC and another phage ST64B, and in the other isolate, we've got a different phage, the RE2010. So we were already able to discriminate these samples after 20 or 30 minutes via this approach of mapping to a database of taxonomic defining genes. So after that, we took the same data, and I built a reference tree from of all Salmonella entericas, and I wanted to see whether or not you could confidently determine whether or not which serovar it was. So I used a mapping approach and then p-placer, and was able to plot the log ml of the chance of that sequence being placed into this clade here, which is the Enteritidis clade. And you can see that after 20-30 minutes, we've got the increasing probability of it being in this red region here. And you can see that that's nicely separating from the quite closely related Colorum and Gallinarum serovars. So, and lastly, we've got someone's printing. Lastly, we want to determine whether or not with the same approach of mapping and p-placer, whether we could determine something was outbreak or non-outbreak. So we were very grateful to have the chance to integrate our data set with the ongoing surveillance sequencing from Collindale, and we were working with those guys. And what we found was that this is the outbreak strain, and this is the probability of it being placed into the outbreak cluster. You can see that after 100 minutes, we've got a high probability of it being within that outbreak cluster. And then and the other non-outbreak strain, you can see it placing elsewhere onto this reference tree. And we've got after 120 minutes, strong probability that it's placed there. So we were showing that after 10 minutes, we were able to speciate these strains, and we were able to detect these chromosomally encoded phages. After 30 minutes, we could determine the serotype, and after 100 minutes, we could split the outbreak from the non-outbreak isolates. So I've got a long list of acknowledgements here. So University of Birmingham, Warwick, everyone at Oxford Nanopore, and all the guys at PHE and Heartland. Thank you.
Very cool.
Clive, what did you think of that then?
Well, I love it. You know, I've seen it before. I love it. You're publishing that, right?
Yeah, we hope so. We've submitted a manuscript. Yeah.
Okay, so I mean, I just see room for improvement. I just want to get the times down, basically, and I'm wondering how we can interface top loading with the sample directly.
Yeah, that's the thing we would love to be able to do. I mean, how quickly with the newer library kits could we get the sample prep down to? Do you think?
Well, I think that it depends. If you're spotting, body fluid onto a flow cell, it's the time taken for the reaction to occur and the DNA to migrate into the flow cell, basically. I've no idea how efficient that will be. We only have proof of concept. Everything basically works, so there's always an efficiency issue. But I think it could be very short. I mean, what I want to get to is near real-time testing, where you take a sample and you touch it to a device, and within some reasonable time, we're talking minutes, you can see what's going on. And I think that's where we have to get to. But this is great. I mean, this is phenomenal.
All right, that's brilliant. So I mean, because Josh is doing a PhD, I thought I'd get a guy to start drilling outside, and I wouldn't put his face on the slides initially, and then I'd ask someone to print remotely on our printer just to distract him. But I think you'll all agree he's done a very good job there. So thanks very much. So we'll try and keep as much time as possible. So I think it's time to go over to Torsten Seemann, who is sitting also in my office, and we're going to have to do a quick change around with the voice here. And Torsten's actually been sitting here while we've been on air, completely changing his talk, writing a new talk. I'm not 100% sure why, but he'll tell us about his new title. It's also been inspired by the proceedings. So just a little change over here with the sound. I'm going to mute. Put your sound up.
Hi everyone.
Just want to check with Clive that you can hear you.
Yep, I can hear you.
That's it. Okay, off to you, Torsten.
Yeah, hi everyone. I'm the token non-Englishman here today. I'm visiting Nick here in Birmingham, and he asked me to talk. My original title was Awesome Microbial Genomics Pipelines, but I sort of didn't quite have anything that I wanted to talk about just yet, and I would have liked to talk about some nanopore data, but as being in Australia, Clive mentioned that there were some shipping issues related to a US postal strike, and our reagents arrived mildly damp, and all we managed to get out was 7 reads of mycobacterium. So we're hoping for better results in the future, and hopefully I'll be able to talk about some nanopore analysis then. But instead today, I am going to put my slides on now and talk about something more general. Are they on? Yep.
Yep.
So I guess one of the things that sort of come out of this nanopore data is Matt's talked about sort of this processing on the fly, and there's a class of algorithms that work with data on the fly that are called online algorithms, and online algorithms are going to require a sort of a whole new approach to things. I mean, everything we're doing currently in genomics is based around batch processing, and one way to make online processing work is to do exploiting parallel programming or parallel computation. So what I thought I'd do is give an overview of parallel computing and how we can use it in bioinformatics. I'm not going to talk about streaming applications specifically, but just to give people an idea of the sort of level of parallelism you can do in bioinformatics. So there's a bit here for people that know nothing about sort of parallel computing, and I think even if you do, there's probably some tidbits at the end which will be useful for your own work. I'll just get the first slide going. So there's kind of 3 main types of parallelism. So most of you will be familiar with clusters of computers. So this is like Amazon, where you're distributing the workload across a bunch of networked computers. So that's sort of the big view. Then if we look at an individual node on a node in that cluster, most nodes these days have multiple cores or multiple CPUs with multiple cores. And so this sort of symmetric multiprocessing is another level of parallelism. And then you can go even further. On a single core, there are special instructions that the CPU runs called SIMD instructions, which allow you to do the same operation on a vector of data at the same time. So you can sort of see there's these multiple levels of parallelism, and the way to get the most performance is to take advantage of all 3 levels. I'll just cover clusters first. So clusters kind of first started 20 years ago or so, where people would just connect PCs over Ethernet, and that is fundamentally today what clusters still are. But obviously, vendors have optimized these and made them very high density, so you can fit lots of them together and have fast interconnects and sort of these blade systems you hear of as what's going on there. And then there's very sort of specialized computers. So the IBM Blue Gene is an example of that. Very high density, which means lots of CPUs and RAM in a very small amount of space. Low power, which also means low heat, so it's cheap to run on electricity. And very fast, multi-dimensional interconnections with low latency. So to take advantage of a cluster, what do? You take your task and you just break it into subtasks. So some tasks are pleasantly parallel or embarrassingly parallel, which is a good situation. You just sort of break your data up, submit each subtask to the cluster queue, and then just combine your results. And sort of taking a large set of sequences and blasting those is against the same databases is sort of a good example of that. But many other tasks, such as de novo genome assembly or other things like that, need to communicate during the run. They can't sort of work independently. They need to. What they do next depends on what's happened so far on other nodes. And there's various ways to sort of do that, and I'll talk about that later. So clusters was sort of the first level of parallelization, and then sort of multi-core symmetric multiprocessing is sort of the middle level. So that's where you have multiple cores on one node. And once again, the simple way to exploit multiple CPUs is if you've got an embarrassingly parallel sort of problem, you can just break it up into subtasks, one per core, run them, and join them. And there's lots of tools in bioinformatics that can exploit multiple cores, these multi-threading tools. So aligners like BWA and Bowtie and even SAM tools now have support for multiple CPUs. And many languages natively support multiple CPUs. So how do we use symmetric multiprocessing if you're sort of, how does it actually get done by the underlying languages? Well, a standard Unix, say macOS or Linux, sort of has this idea of POSIX threads, which is sort of a standardized interface to using multiple threads. And lots of the tools you use that. A more simple interface to that is sort of OpenMP, which is uses sort of hash pragmas inside your C code to sort of help tell the compiler how to parallelize your stuff, and it will then use multiple threads underneath. And finally, without even really knowing it, you are using SMP when you sort of use job control in Unix. So when you put something in the background with an ampersand or use a makefile or GNU parallel, you are actually effectively doing SMP. So like I was saying before, sometimes it's simple and the tasks can just independently do their work, or sometimes they need to talk. So there's all sorts of ways to do this inter-process communication, and there's simple things like just using touch files is one way of sort of communicating. But when you pipe two commands in Unix, you're doing an inter-process communication or using shared memory or all sorts of other things that I won't go into. So the third level of parallelism, we had lots of computers, and an individual computer has multiple CPUs, and then within a CPU there is possible parallelism called SIMD, which not many people are probably aware of. So before I can sort of explain SIMD, I have to give a sort of an introduction to machine code. So CPUs don't actually run your Python scripts directly. The instructions get converted into native machine code, and the machine code is different for an Intel processor as it is with a Spark processor or the old PowerPC processors. But I've got a little example of 4 lines of machine code here. So R0 is a register. It's kind of like something that in the machine stores a value. So let's load into register 0 a variable called years. And then we're going to multiply what's in register 0 by 365. And then we're going to add 1 to it. And then we're going to store it back in a variable called days. So that's basically what machine code looks like. It's very simple atomic operations that do one small thing. And they change one value at a time. And the thing about machine code is that these registers, like R0, they're very fast to access, whereas going back to memory is relatively slow. So we want to try and keep things in registers and out of memory. So that was a nice simple introduction. So let's imagine we want to do a vector dot product, which, given 2 vectors, essentially multiplies the first element in each vector and adds it to the multiple multiplication of the second element in a vector, and so on, given by that maths formula on the slide. Now, how would we do that in sort of, say, a C-like or Pascal-like language? Well, we would have two variables, x and y, which is sort of a vector of 8 integers, and would have a variable called sum to sort of where we're going to store the result as we go. We set the sum to 0, and then we loop through each element in the vector. So sum becomes the old sum plus the product of the ith element in x and the ith element in y. And by the end of that loop, which loops over 8 times because the vector is 8 long, we end up with our dot product. Now, the number of operations we had to do was sort of essentially one operation to set the sum to 0, and then we went through the loop 8 times, which had to do a multiplication and an addition and an assignment. So altogether, we sort of had to do approximately 25 machine code operations. So how can SIMD improve that? So SIMD, instead of operating on scalar registers, i.e. one value at a time, it can operate on sort of multiple elements. And 8 at a time is sort of a common thing. So in SIMD, the previous example would be written as, there would be a register which stores vector values instead of scalar values. So here we're loading a vector register with the contents of some memory at position x. Then we're loading another vector register with the contents of vector y. And now we're doing a vector multiply. So instead of having to sort of loop over, we can essentially just say multiply vector 0 by vector 1, which does the sort of the element-wise dot product component, and then we can use a vector instruction which sums up everything in a vector register, which is v0 now, and puts it into a scalar register. So instead of the 25 or whatever operations it was now, that loop is replaced by a single instruction, which means we only need to do 4 operations. So these instructions have been around for a long time, these vector instructions, going back to MASPAR computers, which Monash University had and was still being used when I was an undergrad, to connection machines and DEC alphas and all these Cray superscalar machines. They're essentially SIMD machines. And all of us have SIMD machines now. So back in the 90s, everyone may remember MMX was introduced on Intel, and that essentially allowed you to had SIMD instructions to work on 8 bytes at a time, or you could configure as 4 words at a time, and so on. But since then, there's been SSE, SSE2, and so on, and now AVX as well. And they all can now, I think you can maybe operate on 64 or even 32 or even 64 pieces of data at once. And these SIMD instructions are being exploited by lots of the tools you use today in bioinformatics. So they're not really accessible from scripting languages. You usually have to use C, but there's plenty of libraries that you use are already exploiting it for you. So those of you who use NumPy, Python, SSE is being used there in some of the core functions. The GNU Scientific Library, which is sort of an underlying library for lots of algebra, matrix algebra stuff you do, and BLAS, all using that to help speed up, especially matrix operations, which it's well suited to. But the key tools you're probably using it for are things like Hammer and FastA, and BWO and Bowtie, which scale beautifully, use it quite well. So you're wondering, well, how can I take advantage of it? And some compilers are smart enough to sort of convert some of your things into SIMD already. They're not great, but they might give you some advantage depending on how you've sort of written your code. So if you want to take advantage of it, well, if you're using the Intel C compiler, it automatically does it. If you're using C or C++, you can use those command line options there I've given on the slide, and it may vectorize some of your loops if you're lucky. So how can you use SMT? If you're not a programmer, you can't access these intrinsics, then you can sort of effectively do it using Unix. So here's an example where, say you had to do something for 23 chromosomes, you could loop over your chromosomes and run BWA to produce a SAM file for each one in the background in parallel. So if you had more than 23 cores on your computer, you could write this loop, spawn them all in the background, and most importantly at the bottom there is the wait, which will ensure that any spawn jobs all complete before the script continues on, where you could then combine the BAMs or SAMs or do whatever you want. Another way to do that more sort of generically is to write a make file. So imagine we've got 3 FASTA files there, here, 1, 2, and 3, we create a makefile where we say we want to create 3 SAMs. We give a rule on how to convert a FASTA and some reads into a SAM, align them, and then to actually get, usually if you just type make, it'll just do one thing at a time. But if you give it the -j parameter, it will use 8 cores and do as many things as it can. It'll try and run 8 jobs at once as long as it can until it gets the result. So after you'd run that, it would use 8 cores and give you your 3 results. Sort of a nicer way to do things now is with the GNU parallel, which is a fancy Perl script which allows you to sort of parallelize generic things in Unix. So imagine again the same thing. We've got 3 FASTA files, and we want to align the reads to them and create SAM files for each of them. And you can do this with the parallel command here. So parallel-j8 means use 8 CPUs, and then we've given it the command. And the braces here sort of get substituted for each of the FASTA files here at the end. The syntax is a bit foreign at first, but it's not hard to get a handle on. I guess there's a few other underused multi-threaded tools under Unix as well. So PIGS is a parallel gzip, and I would recommend everybody use this because it scales beautifully with number of cores. So if your disks are fast enough to write, that is, it'll scale easily to 64 cores in my experience. Now most of the gain is from is in compression rather than decompression. That's due to the sort of the nature of gzip, how it's stored. But yeah, it flies. PBzip2 is the equivalent for bzip2, and even Unix sort, you know, is now parallelized, which most people don't realize. So you can give it a -- parallel option, and it will at each merge stage, it'll sort of each of the things before it merges, it'll sort each of those with a separate core. So that's all simple things you can do to take advantage of all those cores you have now. And of course, these are, if you really want to sort of abstract even further away, there's many pipeline systems out there that you could use, such as Rufus and Rubra, bpipe, Nasoni, and there's probably, I'm noticing, 1 or 2 published every month in the journals. So, as I talked before, when you're doing a pipe, you're actually using SMP without even realizing it. So, if you pipe program A to program B, you're actually spawning A and B at the same time, and B is reading data through the pipe from A's output as you go. And you can pipe together as many things as you want. So here's a simple example where we are going to take the standard Unix dictionary, chop out the first 4 characters, lowercase it all, count, remove duplicates, and count them, and then sort it and find out the top 10. So this is going to find, you know, the frequency distributions of formers in English. The result is that, which was a little bit surprising to me. There you go. Subshells. Trying to speed up to keep us on time. So subshells is something which I only discovered last year, embarrassingly. And I guess the use case is this. So imagine you've got a tool that only accepts FASTQ files, but you've got compressed FASTQ files, and your disks are slow and you've got no space left to temporarily uncompress them. So subshells can help you here. So whereas if you'd run your command like this here with the 2 raw FASTQ files, but now you've got compressed ones, you can actually replace the file name here with this sort of subshell here, which will uncompress it on the fly and feed it straight in. Now, this only works with tools that sort of read files in a streaming fashion. If they need to sort of randomly seek to the end or the middle of the file, this won't work. But this trick, as described here, works for most tools that read FASTQ files. Not only that, you can go a bit crazy and imagine that you can put arbitrary Unix commands in that subshell. So long as their output at the end is what you need, you can use it. So imagine that instead the tool didn't take FASTQ files, it only took FASTA files. So you only, once again, still have compressed FASTQ files. So subshells can actually do whatever they want. So here I'm taking my compressed FASTQ, piping it to paste, which puts 4 lines into 1 line, cutting out the first 2 lines, which is the ID in the sequence, replacing the FASTQ at sign with a greater than sign, so it's FASTA, and then this tr command is converting it back to FASTA format, so putting it from 1 line back into 2 lines. So that's quite convoluted, but it gives you an idea for the power of what can be done with subshells. And you can actually nest subshells. So a subshell can itself contain another subshell recursively. So yeah, at your own risk. So putting it all together, I guess I wanted to sort of show how we could put some of these tricks into a single script. And some of you, I'm sure, are doing this, but others may not be sort of aware of what they can exploit and what they could achieve. So the canonical example is standard example is making BAM. So to make a BAM, we have to align a FASTQ to a reference, use a SAM, convert that to a BAM, sort it, and remove duplicates, and then index. So we could actually do that without any intermediate files whatsoever. So here's a sort of template line you may want to include in your pipelines, where we're going to run BWA to do our paired end alignment to a reference using 16 CPUs. Because BWA outputs a mem outputs a SAM file, we pipe that directly to SAMTools view to convert it to a BAM. Now notice view also takes, SAMTools supports multiple CPUs these days, and we also pipe it as an uncompressed BAM because we don't want to bother compressing it before we sort it, because we're going to just have to uncompress it again. So the minus sign here at the end says put it to standard out. That gets piped to SAMTools sort, once again using the multiple CPUs option of SAMTools sort. And I've bumped up the memory there so that it doesn't write anything to disk. And once again, output that to standard out, remove duplicates, and then save it as an out.bam. So without using any intermediate files, and even if say BWA didn't support gzip, we could have used some subshells there. We could have done everything without any temporary files. So I guess as a summary to this talk, I just wanted to sort of say that at the cluster level, we've got that pretty well solved with pipelines and queuing systems and the like. The SIMD level is probably too low level for most of us, but the key tools that we're using are all like Hammer and BWA and the like, are all taking advantage of SIMD with some impressive programming. But I think the SMP level, so that middle level for multiple cores is where we still have lots of single-threaded bottlenecks. And I think it's crazy not to be using them when we could. So I guess that's one of the themes in the tools I develop, is to be as multi-threaded as possible. And I think it's the only option we have with the data deluge at the moment. And I think even say for the MinIron, I guess if we are going to have a tricorder, it's going to be a low power, the processor on this tricorder is going to be a multi-core low power CPU, just like we have on our smartphones today, and we're going to have to think smarter to take advantage of all those cores. And yeah, I encourage people to use pipes, subshells, and GNU parallel wherever possible. Thanks for listening.
All right, well, thank you, Torsten. So I wasn't really sure where you were going with all that when you were cracking out the machine code, but actually you've given us some really useful little recipes there and some ideas about how to be a bit more efficient. So thank you for that. I guess we're really getting to 4:15, which is when we said we were gonna stop. So I will just go and have a look on Twitter and see if anyone's got any specific questions. Michael Hoffman said you might be able to speed things up with buffer size and GNU sort in some cases.
Yep. GNU sort has buffer sizing options. That would avoid writing temporary files to disk for the merge. That's good advice too. Or multiple threads.
Yeah. Okay. Right, let's have one last little look on Twitter. Okay, so I think what we'll do is we'll just swap over again, Torsten, if that's all right. I'll just go. Muted, your sound off. Okay, hello again. All right, so I think we're coming to the end. I did just want to see if I could do one little thing before we finish, just to keep the nanopore theme going to the end. Because I just wanted to check in on this nanopore run that I promised that I would release the R7.3 data. And if you're interested in seeing what nanopore looks like, this is actually what the Minnow client looks like when it's actually sequencing. And if you're really sad, you can watch the first 2 hours of the MinION run, which I actually streamed when we put this on Google Hangouts the other day. Yeah, some people have looked at that, and so this is coming to the end of 48 hours, and we can see about 435 pores have actually reported at least one read, 42,000 reads, about 358 megs. So Josh probably not going to win anything with this one. Maybe not. And we can see if we switch to the channel panel what the state of the sequencing is. And essentially what we're looking for on this page is green stuff. So green is strand. That's a pore that's actually got a DNA fragment in it at the moment and is actively sequencing. And what you tend to see is at the start of the sequencing you get a couple of hundred, up to a couple of hundred in strand, maybe 300 would be very good going. And that kind of slowly drops off over time. And you can boost that by either adding some new DNA and fuel, or sometimes the software seems to do something which kind of automatically boosts it, which I don't necessarily understand. And then over time, wells become saturated, which means they're just not producing good signal, or zero, which means that there's not a pore detected in that space. And so this one is kind of almost finished with 12 in strand. So I think we put this on nearly 48 hours ago. So when this finishes, I'll pull the data off and add it to that GigaDB repository. And that's the read length histogram. And it's slightly confusing because it's actually counting both the template and complement events there. So the reads are a bit shorter than what appears there. Right. Let's see if I can get back here. Brilliant. Okay, so any final thoughts from any of our participants? Clive, anything you wanted to say?
No, that's all very interesting.
Oh, you're muted. Oh, sorry, hang on. You're probably muted, Clive.
I haven't muted.
Oh yeah, I've got you. Sorry, say again.
Yeah, all very interesting. Love it. I just want us to get your the gap between field performance and internal is the key focus for us to get sorted out. Okay. But at least you're clearly getting some value out of it right now. So if we can get the rest of the map community at least up to your level, I think we'll be in a good place.
Yeah, well, I mean, we're obviously excited about it, and it's obviously doing a good thing. And we just have to. I do think we have to remember that this is a portable single molecule native strand sequencer that plugs into your laptop that hasn't really cost me more than a few $100s once I get my refund back. So I can't really argue with that. And we've already seen today that you can scaffold a whole bacterial genome into one piece, even at this early stage.
Yeah, I'll just say that you asked about pricing.
Oh yeah.
It will be competitive.
It will be competitive.
It will be highly competitive.
But what with?
Well, I think it depends on the device and where it's positioned. But either way, it will be competitive given the value of the data.
Yeah, and what about the idea of having, short? Maybe we don't want a lot. Maybe for some assays we don't need a lot of data, but we want to run a lot of samples.
I mean, I think we can differentiate those flow cells and other things, and then price them on a spectrum that might suit different people differently. I'm sure all that will come once the wrinkles are ironed out. There is a new flow cell. I didn't put it on my skunkworks thing. I have another flow cell and I can't talk about it yet, but it's all that.
I better stop you before you get in trouble, right?
You better stop before I get in trouble.
Okay. All right. Well, so just back to me. So I hope you enjoyed this. First time we did this, it seemed to have gone relatively smoothly. I think we had around 250 people viewing at peak. I suspect that's dropped down since my witterings, but thank you for tuning in. Most, if not all, of the talks will be archived on YouTube, and I think that happens quite quickly. I just edit it and put it on YouTube. So I guess we'll do this again. Seems to have worked well. And thanks for tuning in, and go and have a Balti. That's what we're going to go and do. Okay. Bye.
Bye-bye.
Bye-bye.